Thanks so much for the introduction. Thanks so much for having me here. It's a pleasure to be here. It's actually my first time at JGI, and I'm a big fan of the conference. It's been great. I came with a bit of a cold, so if I sound like I'm a tired, chain-smoking trucker, it's because I kind of feel like one. But I do want to point out there is a study. If you do feel like you're sick, you can actually order kits and get swabs and send them back, and actually Rumi Chinara will tell you what you probably have. And as soon as I get back to New York, I'm going to send mine back in. So just as a quick plug. So I'm going to start and describe a whole lot of things that are happening in the lab. This is going to be more like a movie, but also I tweet all my slides. I'll go back quickly, so if you want, you can download them all. It's going to be a lot of information very fast, but I have a lot of loves, and I want to share them with you today. So, but all the slides will be available. So, and I'll start with something that's a known fact to everyone here in this room, is that we're really on the foothills of the end of a revolution in sequencing, which really arguably represents the fastest technological revolution in human history. So it's an extraordinary time to be a geneticist, to want to sequence things. It's kind of like a euphoria of genetics, actually. And so what's happened is what used to be in 1673, the idea of a microscope being a new way to explore the world, what we have now today are molecular microscopes. And there are many, many kinds of them that we've studied a lot in my lab, and many people in this room have played with a lot of these technologies. And so it's an extraordinarily exciting time because now you can pick a lot of these tools and use them as molecular microscopes to scan the world around us. And so everyone's, of course, very excited about the $1,000 human genome, but we are more than human. As most of you know, in the cellular democracy that is your body, you are a minority party. So if there was an election, you'd probably never win, because about anywhere between 50 to 90% of your cells are bacterial cells. And maybe the active transcripts is probably outnumbered 150 to 1. So I'm going to talk a little bit about the human microbiome. Again, your eukaryotic cells are much bigger. They're kind of fatter. They look like this, you can see. But you're outnumbered usually in most cases. And if you step onto a scale, it's about 3 to 5 pounds of bacteria. And so if in some cases your diet is not working, it might not be your fault. You can clearly blame some of these guys. And we now know from a great work from the Human Microbiome Project that every area of our body is colonized by this ecosystem that really surrounds us and helps mediate health, and now we know even more disease. And so this has led to a large degree of microbiome hope, I'd say. So it's led to many national as well as international consortiums studying the microbiome. Many of you in this room are involved. We can understand obesity much better through the microbiome. We can understand how food gets processed in the body, links to inflammatory bowel disease, links to over a dozen different human diseases are now better understood, even mental diseases, through the lens of the microbiome. So it's a pretty exciting time for sure. And some of my favorite work actually comes from Michael Fishback and Mohammed Dani, where they've been mining public data to look for new biosynthetic gene clusters, which has come up a lot in the last couple of days, of these sort of pathways that actually are driving some of the biology. And in this case, you have organisms that work on your behalf, make sort of this protective microbiome shield that you get just because you have a nice vaginal microbiome. And so they even are similar to what's being developed in some pharma companies. So the big question that happens, and this is getting a bit into my background and excitement about microbiome and metagenomics, is really simply, where do they all come from? We know that as you're developing in utero, the placenta, previously thought to be just a sterile organ, we know that mothers give us a lot beyond just love. Some of you got more love, some of you got less love. I can tell by your looks on your face. But they should have all given you love, as well as a microbiome. In this case, the placenta is its rich ecosystem, and the vaginal microbiome you often inherit at birth. And so in this case, unquestionably, sharing is caring, I like to say. There's another case where sharing can be considered caring, and this is fecal microbiome transplants, which if you haven't heard of these, they are exactly what they sound like, insofar as it's a way to transplant an entire fecal ecosystem into someone who has inflammatory bowel disease or Crohn's disease. And for a little while, people got really excited about this, and still to this day. But the FDA had to basically start to fast-track clinical trials because people started doing it themselves. In this unfortunately titled article from Scientific American, I'd say here, because there were websites popping up, and I couldn't make this up if I tried, something like thepowerofpoop.com, which if you go there, you're like, well, what's this website? So you go there, and it's safe, accessible microbiome transplants for all who need it. People aren't doing this, though, because they're crazy. They're doing it because there's a 95% success rate for C. diff treatments, Clostridium difficile infections, and even treating other sort of gastrointestinal disorders. People started sharing their own microbiomes, banking them, Twitter pictures of people banking their own stool in their own freezers, which I do not recommend you label it meatloaf or something like that and put it in the freezer. People doing this, people putting on Twitter about it, this led Eric Alm to start openbiome.org, which actually is an organization that banks healthy stool, checks it for pathogens, and uses it as a way to transplant fecal microbiomes. Now, they even had a great campaign for a while that was really, they noted, how many people can you treat? They had this to kind of have people care campaign This was a real campaign on Indiegogo, and they said this might be the most important thing you do all day. Like, you throw it away, but this could be medicine, right? And so it's true in this case, because sharing is caring, right? And even if you're hard up for cash, you could make about 13 grand a year, actually, if you're regular, I'd say. So in this case, we laugh about this and joke about it, but it's an extraordinary success rate. Almost nothing in medicine has a 95% success rate. So it's extraordinary. That's why people are excited. But then it does raise really interesting policy questions. So how do you regulate fecal transplants? Is it a tissue or is it a drug? It's actually, from everything we've just told you, it's actually both. And so far they've been regulated more as tissues than drugs, which makes them easier to pass on. So it's been good news in that sense. So that represents, I think, in a nutshell, the microbiome hope. There is, of course, some degree of microbiome fear. You just say the word bacteria and people will freak out in general. You can go to a gas station, you can get gloves if you're afraid of germ-laden pumping, you can get germ-free pumping if you want. Maybe you go to your hotel room, you're like, I'm terrified of this gas station now. So if you go on a vacation, you'll go to your hotel room, you'll find hermetically sealed remotes that are easy to clean because people are afraid they're dirty. So you think, well, I'm terrified of my room. Now maybe I'll go to the pool, take a nice dip in the pool at the hotel, but then you might see a sign in the bathroom. This is from a real sign that I took a picture of once. You think about, like, in this case where things get in your mouth, where sharing is not caring, I'd say. So this represents, the fear people have. But even on the New York City subway, people are afraid to touch the railing. Sometimes they'll put a barrier of essentially a paper towel between their hand and the railing. But I'm more afraid of this paper towel, actually, than of that railing, because it's used, it's nasty. I even caught one woman with a lab glove riding the subway, just terrified to touch the railing. And so this dawned on me about 2.5 years ago that everyone was really afraid of just touching different surfaces. There was all this fear, but whenever possible in life, I'd like to try and replace fear with knowledge. And so I was also inspired to start looking at this question, mainly just because I'm a father. And so this is my daughter. When she got old enough to start riding the subway, I thought, what is actually there? Just what is actually on the pole? And no one knew. There'd been a little bit of work by Norman Pace. There'd been some work on air microbiomes, but nothing about the very basic question of what is in the shared urban ecosystem that we all share, essentially, and transit to each other. So what do you do when you don't know? You explore. So we started collecting swabs all around the city and then built what we call sort of a city-scale metagenomic profile, or what the New Yorker called a metropolome, you could say. And so we published the first of this work last year, sort of the first city-scale metagenome profile, where we went across all of New York City, swabbed every station, and characterized what we found. And so we did this through methods that have been described already at this meeting, where you take all the DNA, we do shotgun sequencing and metagenomic sequencing. I like doing all the DNA because all kingdoms deserve love, so we did shotgun sequencing, mapped all the reads to known reference genomes, and then quantified the abundances. And so in particular, we built an open source app, so as people collect data, you can actually take a picture of it, timestamp, geotag it, and we did extraction using mostly mobile protocol and sequenced over 10 billion fragments on a Illumina sequencer, trimmed them, and then used BLAST and Metaflan to characterize them. And my favorite thing of the study, actually, was that when we mapped all the reads back, we could see about half of the DNA in the world around us matches no known organism. So it was very much a mystery right under our fingertips. But I find this very exciting. I wander around the subway, and I'm excited about the mysteries under my fingertips. Other people think that it's aliens, and I can't, there's no evidence that it's alien DNA. Unless aliens came and left DNA, we would then sequence it. But that's not what we think it is. It's just the lack of, as most of you know, the lack of reference databases. There's so much left we haven't yet sequenced. But the rest of it mostly was bacteria and some viruses and some other kingdoms. I'll go through them in short order. But we did see essentially over 600 species of bacteria. We submitted 656 supplemental HTML5 files where you can zoom in and out of the data sets. The Wall Street Journal also made a nice graphic where you can see where is most of the mozzarella cheese or other sort of cheese-related bacteria floating around the city. You can play with the data set here. We also posted what are the top species. So most of it was Pseudomonas, some staph, some Enterobacter, some Actinobacter. Things that are normally associated with skin was the most abundant species that we found, which is not too surprising. And we did also see that the bacteriophages actually were the top-ranking ones would match often the bacteria. So in some sense, metagenomics is a great way to cross-reference what are the top-ranking organisms you find between kingdoms to see essentially what is a validation of abundance. In this case, the bacteria, we also had a bunch of other tables that listed all of them. We also did some blasting of all the sequences to see what might be there. We found a lot of, we found human DNA, a lot of other French mouse, as well as rodent DNA, as well as some other potential insect and other species. We had one sample that had this huge burst of chickpea and cucumber DNA, and the only reason we can think of is that someone was eating a falafel and then touched a surface, and then we must have swabbed it. So, but the whole blast table's there. All the results are there if you want to mine the data. But we did see, for example, some more plant DNA that was near Central Park than away from it. Some other results like that aren't in the paper. But it's too low sequencing, I think, for a lot of those genomes. But some other things we saw in the data that I liked was that we knew that there would be some human DNA. We didn't know how much, but we know that some days on the New York City subway have more DNA than others. So for example, once a year, there is a no pants subway ride day. We did not swab on that day for this data set, but we do now make sure to match our collections with these days that occur annually because we want to see this spike in human DNA. Just to be clear, by the way, it's not no underwear, it's just no pants. So it's actually fairly respectable, I'd say, as a social activity, whatever that could be. But so I think in this case, we actually, and I'm going through the data set a bit quickly because this is all published for about a year, but we actually mined the data that's, we essentially predicted ancestry informative markers for human DNA, mapped it back to the human genome, and then compared it to the census data. And you can see here, here's the prediction from Asian, Black, White, and Hispanic ancestries versus what was calculated the ancestry, between the census data and our sequence data. In different areas and neighborhoods of New York City, we could predict what are the likely DNA ancestries just based on the human DNA left behind. So that actually means that when all of you leave this room, we're going to have a team come through and sequence the DNA on all the chairs and give you a report back and then tell you about your family history and where you grew up. And I'm kidding, we're not going to do that. But we could do that to some degree. And so it's extraordinary. It's the first example of a city, ability within a city to track essentially what the DNA is that was left behind on city surfaces. So the other thing that we did in the data set, this is more, once it got published, we've been mining it since then. And so this is work with Mohammed Dani. We've been actually trying to find new biosynthetic gene clusters. Can we actually dig deep in the data and find new BGCs, essentially prediction of essentially functional elements that we could find from these bacteria. And so in particular, we've been mining even bacteria that are on steel railings in the subway system. And this has been predictions of some of the small molecules that might be made from this abandoned subway station that we got access to with the MTA. And you can see here that things that are just living on steel railings seem to be potentially producing some of these small molecules, which Mohammed's been working on validating in his lab. And so this is also some of the continuing work we're doing with basically all of the data sets as we keep going. The other thing to note about the data set is that species diversity did differ depending on where you were in the city. This is some of the example files we submitted with the paper where there's a density map we made for each organism across the city. And we found some differing densities, but we also saw something really peculiar. In this one area, there was a burst essentially of Shonella bacteria and also some Pseudoaltomonas. In this case, that was the one station that was flooded by Hurricane Sandy. And it turns out that there were 10 species of bacteria that we didn't find anywhere else in the subway station, and we haven't actually seen them since unless we look at other sort of water-related samples. And so in this case, it represents this kind of molecular echo of what had happened at that station. Sort of the cold ocean water had been left on the walls, and when we swabbed them and sequenced them, we could see species like Shonella frigida, which was previously thought to be an Antarctic species, so first found in cold Antarctic waters. And it actually makes an interesting acid called eicosapentaenoic acid, or EPA, which when I started digging in the literature, I thought, well, what's this? It's normally actually something you get when you've been eating fish. And so, oh, that's kind of cool. What else does it do? If you have low levels of it in your diet, you actually had a higher risk for suicide. So this made me think, well, in a worst case scenario, if you happen to get something from the subway in your mouth, this might not be so bad because it's kind of a helpful bacteria. There's some, Pseudomonas putida that's in there. It's all pretty good stuff. And so one time a reporter had actually asked me and said, well, Dr. Mason, if I were to lick a subway pole, would I be okay? And I say, well, licking is different from touching, and I wouldn't necessarily recommend it. But there are some mostly inert things. You'd probably be okay. It's not recommended, but I think you'd probably be okay. So then, of course, the next morning, this is what appears in the Gothamist. And so, I like to point this out is that the reminder that people will quote you directly, even only 2 words. So it's still true. So I stand by this because 2 weeks after our paper came out, the New England Journal of Medicine showed that actually the worst thing to do if you have an allergy is to take away all instances of that allergy. Your immune system needs training. This study was just validated 2 weeks ago also by another group, and so also an NEJM. And so we do know that you need this exposure. Your immune system needs target practice. So I do still stand by that to some degree. And so we'll come back to the Gothamist in a little bit. So what are we doing now? So that's a little bit of what we've done. That's in the subway. So what's happening now? Next steps are to try to look at the design differences between other subways around the world. New York City is actually only number 7 on the list of busiest subways in the world, so I started a quest after about a year ago to say, well, there's only 1.7 billion riders in New York City every year. What else? What about the other cities? What's happening there? And so we've just planned and launched actually for June 21st this year to have a global swabbing festival. It's across all the cities, all the 10 busiest subway systems in the world, plus 40 other ones around the world, sort of this large-scale consortium called Metasub, which is to basically do three things. We're going to build more geospatial metagenomic maps in cities. We're going to look for antimicrobial resistance markers and also look for new biosynthetic gene clusters. And so you can go to the website and you can see the map. You're welcome to join if you're interested as well, if you want to be a hub city. And you can see where we're all swabbing. And also the reason we chose June 21st was to match the Global Ocean Sampling Day group that's also doing it on the summer solstice. So we're going to coordinate efforts and have this all happen, the sampling occur at the same day, sort of a genetic Polaroid of what's happening in cities and in nearby oceans. The other thing we're doing this summer is actually doing a little bit of fun with the Olympics. The Olympiome will be collected this summer. We're going to see before, during, and after the Olympics to see what happens when 1 million people converge on a city and sort of disrupt the microbiome there as well. So other things that we're doing as part of the consortium is to work with new technology. So we've been working a bit with DropSeq and 10x Genomics to do metagenomic profiling. This is very new developments in the last few months, but we do know that it can, to some degree, work on DropSeq and is likely going to work on 10x Genomics. And of course, doing more work on PacBio to look for some peculiar DNA-based modifications. And the third thing we're doing as a consortium is also partnering with the Extreme Microbiome Project, the XMP, is to try and think of better ways to extract. Our recurrent theme in this meeting has been how do you get all the DNA and all the RNA out of your sample? It's actually a really hard problem. We all know that everything that exists today is pretty much imperfect. And so we've actually been working with Millipore Sigma. This will be released probably later this summer as a multi-enzyme cocktail. Basically has a variety of enzymes that can break apart almost any kind of cell wall. So achromopeptidase, metanolysin, chitinase, lidocase. We're probably going to add cellulase, but not yet in this first iteration. So the idea is to have a freely accessible, or not free, but freely accessible, essentially multi-enzyme cocktail that should be coming out this summer, which is work with the ABRF and the XMP. And so the XMP is my lead into the extreme sites and Superfund sites. And so we also have a project called the Extreme Microbiome Project, which is trying to find really peculiar sites around the world, and sometimes in concert with colleagues at NASA, or also with Scott Tye in Vermont. And so this includes deep ocean brine lakes, or high acidity saline lakes, or even candy pink lakes in Australia, like Lake Hillier. It actually looks like this. It's an extraordinarily pink lake. And so we actually have some sequence data that's posted in BaseSpace right now as well, where you can mine the data if you want. And one other place that we've been looking, which is one of the subjects for today, is of course to look at extremophiles in New York City. We just released a video about sort of the project recently. And so Superfund sites, if you're not familiar with them, are where things have gone wrong with humanity, I would say, where we decided let's just dump all the crap here kind of thought. And then what happens after a while, of course, is the area gets very polluted. So the EPA has a Superfund program to go in essentially and protect human health and clean up the polluted sites. And you might think, well, how many of those are there? There's not that many. There's maybe 5, maybe 10. Well, no, actually, there's like hundreds of them. And so it's terrifying to think about how many sites have been deeply polluted that the EPA considers toxic sites that are slated for potential Superfund sites, including a couple nearby and even in New York City. Most notably, the one that we're looking at is the Gowanus Canal. And so if you have not heard it, it's basically this murky canal where people have articles that are like, we don't even know what's really in there. People are afraid of it, but yet fascinated by it. It's kind of like that ugly cousin that always comes to reunion, and you're like, who is that person? And you don't even know if they're related to you, but they always have interesting things they're doing, and they give you free candy. That's just my family reunion. But even one guy said, well, I'm going to go to the canal, and I'm going to swim in this canal. This is what it looks like, because it was for Earth Day. He wanted to demonstrate how toxic it was. Put on a hazmat suit, and he couldn't make it the way down the canal. He just concluded, again on the Gothamist, it's not safe to swim in there, is what he said, because it smelled like sewage, because it's full of actual sewage and toxins. And so, so what is the canal? Where did it come from? Why did it get polluted? So it's really, when the Dutch settled New York, it used to be kind of a trading post. This is back in the 1700s. Over time, it was built up. You can see here, sort of, by 1849, it was even more built up. You can see this kind of the New York harbor. And then eventually it was dredged to make a canal because you wanted to have even greater shipping. And so tanneries were there. People were doing processing of essentially toxic industrial solvents. And so eventually the water started to look like this. And so now in 2016, this is what the city looks like. And what we did is we wanted to collect different samples from around the canal and get a sense of what was actually there when we sequence it and see what we could find. So this involves a long tube. You've all probably done this before with essentially when you have a straw and like a glass of soda and you put your finger on the top and you pull it up. It's the same thing. That's all we're doing with a big long tube, putting it in the bottom of the sludge and pulling it up and undoing the cap and then taking a sample. And so this is work done, actually one of my favorite things about this project is work done with Nelson Bertholtz, which is a landscape architect firm, plus GenSpace, a community laboratory with Alan Jorgensen, and bringing them back to sludge back to sequence them and profile them. And so the big question is, okay, well, what's in there? What are they doing? So we made this map essentially using a lot with metaflan analysis to see the distribution across all the 14 sites and what are the recurrent themes of bacteria that are found across viruses, archaea, and bacteria. And we can see desulfobacteria actually are persistent across every sample that are essentially these sulfate-reducing bacteria. We could see part of the smell is probably coming from methanobacteria. You can see here, often found in the human gut, of course. And then we did see a lot of Pseudomonas and Pseudomonas petita, which actually might be digesting the organic solvents. And when we did an analysis with human, which is to see what are the aerobic or non-aerobic respiration pathways, it's strongly enriched for anaerobic pathways. You can see here across all the different sites we've collected what's been enriched for anaerobic pathways, which makes sense because it's actually in the sludge is where these samples came from. And the other kind of cool thing I like a lot about the Human2 pipeline is it lets you try and predict what are the bioremediation pathways that are active in these samples. And so we've actually seen that this looks a lot like toluene processing, arsenic, or even coal tar, essentially leftovers. You can see the signature of the toxic waste is being processed by the bacteria that are left. And so what this has prompted us to do is start to think, well, these are extraordinary pathways. So much this rich ecosystem that in the next 10 years is going to be completely dredged, capped, and then filled with new water. But what if we keep these essentially positive bioremediation pathways? And so this got us thinking more about designing the metagenomics and trying to keep some of the good actors around to get rid of the bad actors. And so I would even argue that our understanding of biology is measured by how well you can engineer it and then predict the outcome. And so to this end, we've actually started a course at Cornell called the How to Grow Almost Anything course, which in concert with George Church, which is basically that you can actually do synthetic genomics and modified microbiomes as part of the course. And so med students take this course at Cornell. So we wanted to try and kind of start to hack the information, hacking kind of in good ways, like gain access to hidden information and reconstruct something to derive into use. So we're actually inspired by, as part of this course, the How to Grow Almost Anything course, what if we take inspiration a little bit from nature? Actually, like sponges, for example, have some way to maintain a microbiome, but yet allow passive water to move through. So we thought, well, let's design essentially an artificial sponge that we could try and put into the canal. And so we're inspired very much by sponge sort of shapes and figures, converted this to processing language to design it, and then you loaded this into a Blender, which is a 3D printing optimization software. So you can actually, a lot of these tools are all freely available, all of it's all on our website as well, so you can actually take the CAD file and download it, and if you have a Prusa i3 printer, you can just load it in and start to print your 3D sort of artificial sponges. So this is what it looks like when it came out. And we submitted this design to something called Gowanus by Design, which is sort of a design contest of should we actually redesign New York City. So we pitched to the design firm. We said, okay, well, what if we actually built these essentially colored poles that would represent the collection sites I just showed you, and actually embed in the bottom of them essentially some of these beneficial sort of bioremediation pathway bacteria, and then we could monitor, essentially have data come out as well as the light change depending on the activity of the pathways. And essentially have a sort of a teaching lab over the canal, and for no reason other than we thought it'd be fun to have people kayaking next to watching movies. Well, I don't know, why not, right? So, and so the idea was actually to build this entire infrastructure so that would monitor what's underneath the silt, because eventually the canal will be capped, and essentially it's going to be clean, but the hope is that they'll keep any of the toxic sludge hidden away. But we actually thought, well, what if when we do this sampling, we almost can kind of inoculate and then memorialize and sort of keep track of what's happening to the sludge underneath the cap over the next 10, 20, or 30 years. And so we actually won the design competition. That's currently kind of the plan for a project we call the Brooklyn Bioreactor. And you can actually go to the website and mine all the data that we've sequenced so far. You can you can mine it either by species that are present or by what pathways are active in the sort of metagenome samples that we put in here. And so it's a very, it's a weird project because it's a medical college, a GenSpace, a community laboratory, a landscape architect firm, and then some other designers, and we all get together and just start planning out what it should look like. So it's a very kind of fun way to visualize what's happening in sort of the invisible city. So in the last section of the talk, talk a little bit about what was promised, like at space stations. This is more work we've just started, but it's something that I'm very excited about, is thinking about sort of space metagenomics. And so why space? Because things do grow in space. Of course, this is an example of what mold that can actually grow on and in the space station. So you might think of it as the sterile environment where not many things happen, but it is indeed a place where things grow. And so It looks at what it looks like on the inside of the space station. You can see here things can grow kind of anywhere. There is no up in the space station. It just is all around you. And this is, we do get some microbiome samples. You can see here, if you thought you had a bad toilet at home, I promise you it's not as hard to use as this one. But we've started a project which is basically sort of participatory medicine with twin astronauts. This is Mark and Scott Kelly. Scott Kelly, who just got back to Earth a few weeks ago. And we have a grant with NASA to look at real sort of longitudinal integrative systems biology and try and think about how we could look at their sort of biome and microbiome and how it interacts with others around them. This is the patch for the mission. I'm very excited because I have a patch. It's kind of all I've ever wanted since I was like 12, so now I have a patch. And so the launch happened about just over a year ago. Scott Kelly went in space, and the idea was keep his twin brother on Earth, and then we monitor them both. We look at genetic mutations, we look at epigenetic changes, hydroxymethylation, DNA methylation, RNA expression, and RNA methylation, proteomic changes, chromatin changes, antibody titers, B cells, T cells, telomere length changes, cytokines, targeted global metabolomics, and microbiome and cognition and vascular changes. And we do this essentially about every 30 to 40 days for 6 months pre-flight as well as 12 months in space, and then 6 months upon his return is the plan to build essentially a 19-time point study, including 3 vaccinations, and today we are right here. So the way you can actually study all this data is really have 2 options for sort of space genomics, I'd say. You can either bring them back frozen, which we've done for some, or we can actually do ambient blood draws and get blood basically drop back down to Earth, and we can get it to Houston in about 36 hours. So some of them, when they have been ambient blood draws, they have actually been dropped in the Soyuz, splashed down into the ocean, and brought back to Houston for cell separation. And we can see these are human blood cells that were just not that long before in space. So I was very excited that we actually got really good cells. Human cells didn't seem to mind that much dropping from space, getting picked up by a helicopter and then sorted again. The yield was actually quite good. I was very surprised. I thought they'd all be dead. So we're in the middle of this project now for the twin study in the last 6 months of the collection, but we did have some other work that we have been preparing for, is to actually not just do bringing samples back to Earth, but also do some sequencing in space. And so the reason we're doing this is there's a project at NASA called the Biomolecule Sequencer. This is essentially with Aaron Burton and Sarah Castro-Wallace at NASA. So we're planning for launch this year, June 2016, to send up to space a MinION sequencer, which I think most of you have heard about or read about. If you're not familiar with it, the principle is that you essentially have application-specific integrated circuit into the membrane that measures the conductance change as DNA transits through the pore and gives you a raw current that looks like this. So you can measure picoamp changes and actually see what the trace of DNA is as measured sort of by this song of DNA, you could say. And so we had an opportunity last fall because Andy Feinberg is also one of the Twins Project PIs. He said, hey, I'm going up in the parabolic flight simulator, you mean the Vomit Comet, and I'm going to go up there, and does anyone want to send samples? I said, well, Andy, we should send you a MinION. Let's prepare for the launch that's happening this year. This was last October, and let's see if it'll actually work. Can you do pipetting in 0 gravity? Can you load a MinION flow cell in 0 gravity? He said, sure, we'll send them over there. So this is a video of Andy actually loading, essentially, pipettes, trying to get things moving. Let's see if we have sound here. So the problem in 0 gravity, of course, is that your tips don't really stay where they should go. They kind of float around. So we're designing new tip boxes that maintain the tips, like say, in place. A lot of you, when you do pipetting, you do the double tap, and that you can't do that. It'll all just float away. You can see the tubes float around. And then when you get to the bottom of the flight simulation, they all drop down. So it's preposterous, so you can't help but laugh. So, but we did note it did work insofar as we actually got 3 reads that worked that actually went through the pore, and we confirmed based on the timestamp of the flight that it was actually in 0 gravity as DNA moved through the pore. So we do know that it's possible to sequence in 0 gravity, and so we also posted this on bioRxiv as well as the raw data. So plus an algorithm for mining that data. You can go take a look at it. So in the closing sort of minute of time I've got, I want to say that we're thinking long term. I guess the 500-year plan is still something we're thinking about. I'm excited because at Weill Cornell you can now actually get an area of concentration as a med student in space genetics and technology. And we're working as part of the Consortium for Space Genetics, which is at Harvard, to start to get people thinking long term. And so I think the reason we're doing this is because NASA wants to go to Mars by the year 2035 approximately. And to do this will require a lot of science and technology coming together. And so if you think of really what this means, this actually is not, people like going to Mars and people think about terraforming Mars, but like, oh, you're crazy, we're never going to go terraform Mars. But I'll remind you, Mars would actually be the 2nd planet that we will have engineered at a planetary scale. We're already trying to mitigate the effects of climate change and monitor Earth as many people in this room are, as sort of a global community. And so I think Mars would just be the 2nd time that we will have done that. So in the end, I want to thank all the people in the lab who really are inspiring and make all of this possible. and are fantastic. I think the best lab ever, but I'm biased. I also want to thank Scott Kelly and the other investigators that were just in the mission. This is literally the official NASA poster. They got to pick what they look like. You can see there's the Death Star in the background. They got to pick. And then also want to thank all the members of the Brooklyn Bioreactor team. You can see here Matt and Ian, especially Elizabeth, who's a postdoc in the lab, and also a lot of friends and colleagues who've helped collect, and of course funding from the NIH and Sloan Foundation, NASA, and many other sources. And thank you for your time.
I don't know how it happened. You spoke so fast. You still have 25 minutes left. The questions for Chris? Tom.
Oh, have you seen much difference in the twins? Yet we have not commented on the genetic differences yet because of privacy, but we can see gene expression changes and some microbiome shifts that still maintain within them. They're still distinct, but we see shifts already. And we're looking now. They just came back, so we're going to see what happened after the full year, epigenetically as well as otherwise.